Welcome, everyone. Thank you for joining us for today's webinar, From Start to Finish, Bio-Techne Solutions for Bioprocessing. It is presented by Chris Heger, PhD, Manager of Application Science at ProteinSimple, which is a Bio-Techne brand. My name is Judy O'Rourke, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Bio-Techne. For more information about our sponsor, visit www.biotechne.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Heger. I will now turn the presentation over to him.
Thank you. This is Chris Heger, PhD, Senior Manager of Application Sciences, and today I'm here to talk to you about Bio-Techne's solutions for bioprocessing from start to finish. So let's start with a brief set of terminology. So what is bioprocessing? Bioprocessing is a process that uses a mammalian cell line or bacteria to generate a biotherapeutic protein drug. So what's a biotherapeutic protein drug? It's a number of different proteins, including hormones, growth factors, as well as anti-cancer proteins. Most people are more familiar with monoclonal antibodies and antibody drug conjugates when they think about biotherapeutics. But this also does include both reference materials as well as the widely growing field of biosimilars, and finally, vaccines. Shown here is a typical small molecule manufacturing scheme, in this case for aspirin. It looks pretty complex. There are a lot of reactors, vessels, and steps to make a small molecule like aspirin. Biologics bring significant more complexity. Shown here is a relative size comparison between aspirin and a monoclonal antibody. The antibody is approximately 800 times the size of aspirin and is a protein. Proteins have post-translational modifications, such as glycosylation, that impart a significant potential for variation between batches of product if the process is not tightly controlled at all stages. Biotherapeutics save and improve lives. As the pace of approvals increase, so does the pressure on makers of these new biologics to complete their development processes faster and more efficiently. So when we look at what the bioprocessing industry needs, it's improvements in their production efficiency, meaning more speed, better accuracy, through the use of better technologies. And they also need to have a robust supply chain support so that they can continually make their product. Of course, at the same time, the industry is always interested in means of cost reduction and ways to get to market faster for their given products. Bio-Techne brands offer the complete solution for bioprocessing from start to finish. Our brands can provide cell culture media, proteins, a wide variety of antibodies, small molecules, a large number of different immunoassays as well as functional assays, instrumentation, as well as a variety of different custom services, as we will discuss today. In addition to having the complete solution, there are a number of key benefits of choosing Bio-Techne to support your bioprocessing workflow. You have access to all products from all brands, including some non-catalog and developmental materials, historical and current production records for all of these materials, the flexibility to develop molecules and assays for downstream requirements. We always have regular communication between groups during the product development. We have a wide selection of GMP grade molecules and instrumentation that is 21 CFR Part 11 compliant. And finally, coordination of bulk orders across our brands and product lines is important, And all of this is designed to help build a more efficient and reliable process so that you can get better results in the long run. We have a variety of reagents from recombinant proteins, including enzymes, growth factors, and cytokines. We are an industry leader in providing high-quality antibodies with the capability to develop custom antibodies based on specific epitope requirements and can even generate recombinant versions of your antibodies. In addition, we can provide small molecules and peptides required for various stages of your workflow. We are a leader in immunoassays with our Quantikine ELISA kits, our multi-analyte detection platforms, including both Luminex and Simpleplex platforms, and we can also support custom immunoassay needs. We also have cell-based bioassays and binding assays to verify the protein's activity, and we can also make bioassays customized to your needs. Lastly, we also have a large panel of enzymatic assays covering everything from glycosylation to phosphorylation. And again, in this instance, we can develop custom enzymatic assays to specifically suit your needs. Speaking a little bit more about our custom services, we have extensive capabilities in both reagent and cell culture media generation and the development of both immuno and bioassays. Bio-Techne can provide bulk quantities, custom formulations, all with the assurance that we have sufficient production capacity and we are a reliable supply chain. Our custom services are run by experienced, dedicated product managers and expert scientists. The custom team can provide project consultations before you get started. And once the project is underway, we provide documented milestones and communicate throughout the entire length of the project. So in addition to that final product we aim to produce at the end, we also provide comprehensive reports on the custom product to ensure we've met all of the required deliverables. Through our ProteinSimple brand, we offer a wide variety of analytical instrument platforms to support various stages of the bioprocess workflow. Here I'll introduce them, and later I will show you how they can help solve specific challenges within the bioprocessing workflow. Simple Western is an automated, multi-capillary-based Western blot capable of separating proteins by either size or isoelectric focusing. By using antibody-based detection, Simple Western can analyze complex sample types like cell lysates. An immunoassay readout is achieved by either fluorescence or chemiluminescence on these platforms. Our ICE and Maurice platform is an automated single capillary in a cartridge platform that utilizes whole column direct detection by either absorbance or native fluorescence for our isoelectric focusing. This ICIF assay can run up to 100 samples per run with separations between 6 and 10 minutes per sample. In addition, Maurice can run CESDS, which allows you to measure the purity of your final product. These runs take between 25 and 35 minutes per sample and can run up to 48 samples per batch. Moving on, we also have a single-cell Western instrument named Milo shown here that performs microscale immunoassays in 1,000 cells in parallel. Milo quickly separates proteins from single-cell lysates and allows for multiplexing of up to 12 targets when paired with the InnoScan microarray scanner. Our last immunoassay platform, Simpleplex, features our Ella instrument that takes the headaches out of traditional ELISAs by completely automating the process. Simply add samples and buffer to the cartridge, put it inside of the instrument, and hit go. Ella can process up to 72 samples in 1 hour, generating data with both impressive dynamic ranges and sub-picogram sensitivity. Particulates are never a good thing, whether they show up during the process or in the final product. Our MFI platform provides an image-based analysis of subvisible particles to provide not only particle count, but also morphological information. The MFI can differentiate between protein aggregates, silicone oil droplets, and even air bubbles. The MFI can run up to 90 samples per run and has one of the highest sampling efficiencies of any instrument in the particle analysis field. More sampling means more statistical accuracy and better confidence in your data. Now that I've provided a brief introduction to Bio-Techne's offerings, let's take a look at the bioprocessing workflow and how Bio-Techne can help improve it for you. Shown here are the 5 major steps on bioprocessing, starting with clone selection and cell culture development, product characterization, followed by formulation and stability testing, and final product characterization. These stages can typically be grouped into upstream as well as downstream steps. Let's take a closer look at each stage and how we've helped key customers along the way. Scientists in vaccine analytical development at Merck Research Labs in West Point use Simple Western size assays to screen clones for their molecules of interest. The binary toxin B has a molecular weight of around 95 kilodaltons and is one of 4 protein antigens in the Clostridium difficile vaccine. The Merck scientists screened 7 different clones generated with baculovirus constructs labeled C1 through C7 in the picture on the right, and were analyzed for both yield and product quality. Clone 4 did not express any product, while clones 2 and 3 expressed product, but at a lower than expected molecular weight, suggesting that the protein was degraded and intact protein was not present. Proteins at higher and lower molecular weights were detected in clones 5, 6, and 7, indicating increasing aggregation and or degradation of the product. Clone 1 expressed the protein at the expected molecular weight with no degradation or aggregation observed, making it the clear choice to move forward with for further development. Western blotting technologies, including Simple Western, provide an average protein expression understanding of a given sample. Flow cytometry measures single-cell heterogeneity. However, results are often confounded by off-target binding or variants of isoforms that are bound by the same antibody. In contrast, Milo performs single-cell Western by running SDS page separations on approximately 1,000 single-cell lysates in parallel and can distinguish off-target binding events and protein variants and isoforms, enabling verification of clone monoclonality for the first time. In the example shown, a few wells within the Milo chip have been zoomed in on to illustrate the power of the technology. In most of the single cells analyzed, a single protein in green and a second protein in blue are observed. However, in the last lane, a smaller variant of the green protein is observed, indicating that the cell population applied to the single-cell Western chip is not homogeneous. This would cause concern and potentially have to go back in the process to ensure that homogeneous cell clones were produced. Once the right clone has been chosen to move forward with, we can support the growth of the clone with our growth and production factors. We currently have approximately 50 GMP-grade proteins, including LR3-IGF-1, FGF-basic, and EGF. And if additional cellular pathways need to be targeted while growing these clones, we can provide also ion channel blockers as well as hormones. And lastly, to limit any detrimental impact of the introduced factor, we offer both blocking as well as neutralizing antibodies. We are also making a big splash in cell therapy arena these days with our critical GMP-grade small molecules and proteins to give confidence in providing top-notch quality in this rapidly expanding field. To grow up your clones, you're also going to need media and supplements, and we have you covered there as well. Specifically, we have specialty serum-free media, including chemically defined media, that can be made at a GMP-grade level. We have a wide variety of antibiotics and antifungal agents, often required for culture maintenance. And for your larger projects, specific lots of serum can be reserved and matched to ensure the highest level of consistency. Because not all cells grow floating in media, we also sell basement membrane matrices and purified extracellular matrix proteins. Now let's take a look at how we can help monitor the cell culture development process using Simple Western. Here we are looking at the assessment of antibody integrity in cell culture samples taken between days 6 and 14, both under non-reducing and reducing conditions. Based on these data, the culture can be pulled at the appropriate day for subsequent processing. If we take a look at these Simple Western images, it appears that day 14 is the best, with the most protein being generated. However, if we now look at the charge heterogeneity of the same antibody samples shown on the right, it becomes apparent that the material is changing over this time course. Specifically, the amount of acidic species is increasing while the main and basic variants are decreasing. Knowing the complete picture is much more informative, as waiting until day 14 will actually produce a less desirable antibody product. As we move down into product characterization, the next step is typically purification. Shown here with Simple Western is an analysis of a purification by both total protein, shown on the left with the blue, and immunoassay shown on the right. Since the Simple Western instruments are automated and fast, quick decisions can be made about which fractions to include for pooling and subsequent polishing steps. Once the product has been cleaned up, it can also easily be assessed for purity by Maurice using our CESDS mode. Shown here is an antibody under both reduced and non-reducing conditions on the left and right, respectively. Well, it's the new year and we've got something exciting to help bring it in. Launching next month is our CESDS Plus platform, which aims to provide superior data consistency with a higher number of injections per cartridge, all while offering less protein fragmentation. Please reach out to your local Bio-Techne rep or visit our website for more information. Once you've got a relatively pure product, assessing its activity is next on your list. With SimplePlex, we offer sensitivity, great linearity, as well as dynamic range, and we have factory calibrated standard curves built into our cartridges so you don't have to run them. On the right are two examples for readouts of growth factor and PDL1. Both assays have sensitivity in the low picogram range with excellent dynamic range. Remember, these results are generated in an hour with very little hands-on time, which is way more efficient than using any other ELISA-like technology on the market. Outside of our instrumentation, Bio-Techne also has a wide variety of bioactivity assays to help you understand your product. We have cell proliferation and adhesion assays, apoptotic assays, and many more. We can perform assays for you if we use them to qualify our GMP-grade proteins, and we can develop and transfer dozens of other assays for you to run at your facilities around the world. Shown below are two examples looking at the inhibition of proliferation by either a neutralizing compound to TSLP on the left or anti-VEGF on the right. We also offer a number of binding assays, and we can help develop custom assays to help test binding of your product. To highlight it as an example, let's look at two of our anti-idiotype antibodies. Our anti-idiotypes are brand new, and we have them for many of the most popular therapeutic molecules. Shown here are examples of the anti-idiotypes for adalimumab on the left and cetuximab on the right. These anti-idiotype antibodies were screened against a number of other biotherapeutic proteins, and as you can see, they specifically recognize their intended target and have minimal cross-reactivity with other therapeutic proteins tested. Not seeing your favorite antibody here? Well, reach out to your local sales rep and get that conversation started. We're happy to help along this path as well. Okay, once you've characterized your product for activity and other aspects that we've already discussed, the appropriate formulation and stability of the product needs to be assessed. Simple Western can be used to look at purified samples as well, and researchers at BMS used it to assess heat-induced fragmentation of a monoclonal antibody over a 3-month treatment at 40 degrees C. Increased appearance of a 110 kilodalton fragment can easily be quantified, and an accurate decision can be made on how best to store the material. Similarly, Maurice IEF can be used to look at VLPs, or virus-like protein particles, and the best buffer to keep them stable. Shown on the right are 3 formulations, each with 3 different freeze-thaw cycles. In this example, formulation A is the best for the VLPs, giving the most consistent data across various freeze-thaw cycles. In either case, our platforms can help look directly at the product and how it behaves in various formulations and under various stress conditions. An important aspect to understanding of the formulation and stability is whether the formulation or storage temperature impacts the amount of protein aggregates observed. These aggregates are undesirable if introduced into a patient. Our MFI technology can easily compare aggregate numbers in samples produced with different formulations, as shown below. Formulation C in this case produces the fewest number of aggregates and would be the best case moving forward. Lastly, the final product needs to be analyzed for a number of key attributes. Bio-Techne solutions can help with all aspects of final protein product characterization, including biochemical assays, functional assays. We can help check for acceptably low levels of processed residuals like host cell proteins, and again, check for particulates as well as protein aggregates. Now let's look at some final products. Maurice and Maurice are great for looking at a final, pure product. Shown to the right is an example of an antibody drug conjugate. The ratio of unconjugated antibody to conjugated species that appear to the left of that named peak can easily be quantified. When looking at this class of molecules, the amount of drug payload on the antibody is quite important to understand as it directly relates to the efficacy of the drug. And shown below is another final product characterization, this time on Simple Western for the polysaccharide conjugated CRIM197 protein found in the 15-valent PC vaccine. These data help show the level of conjugated sugar to the protein, which is critical for the vaccine's efficacy. In addition to checking the final product, our MFI technology allows you to analyze the sample for any additional unexpected contaminating subvisible particulates in the storage container. MFI can see silicone oil, air bubbles, as well as protein, and easily distinguish between them. And for the unexpected process impurities like residual host cell proteins, turn to Ella to help out. And with that, I'd like to introduce our second new product and announcement of the day. We are launching a brand new cartridge that opens Ella up to whatever antibodies you'd like to pair into your automated sandwiched immunoassay. Everyone has their own host cells that they use for growing up their clones, and specific host cell proteins are required to be monitored along the way. With this new open cartridge, researchers will be able to generate an assay on their own terms with their own specific CHO-HCP antibodies, in this example, resulting in an assay with great sensitivity and dynamic range. We provide everything that's needed to be confident in releasing a product with sufficiently low levels of process-related impurities. I would like to conclude by letting you know that Bio-Techne can perform many analytical services in-house as well to characterize your protein biologics. We assay samples on state-of-the-art instrumentation using validated SOPs and provide detailed reports for assay documentation. Shown here is a list of the services as well as the instrumentation that we have available for this testing. With that, I'd like to thank you for your attention today and hope that you've enjoyed this webinar. Please visit our website for more information on any of the bioprocessing solutions that we have discussed. We are here to help you get your patients better medicine faster. Thank you.
Thank you, Dr. Heger, for that informative presentation. I would also like to thank Bio-Techne for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through April 30th, 2019. As a final reminder, our speaker will follow up with any questions you submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.